I'm so glad you found your way here tonight. Right now, while you rest, your brain is actively working to lower the very cortisol levels that kept you tense all day. Tonight, you'll discover what stress is really doing inside your mind and body, and understand why simply knowing can help you begin to let it go. Welcome to Sleepy Mind Science. Wherever you are right now, lying still, your body settling into whatever surface holds you. Allow yourself to soften just a little. Tonight we'll journey through 100 science facts about stress. From the first signal that fires in your nervous system, to the quiet chemistry that calms it, to the moment your body finally remembers how to release what it's been carrying. This channel exists for the nights when rest feels far away, when the mind is full and the body is tired, but sleep won't come. Subscribe, and let Sleepy Mind Science be there for those nights. A new video waiting whenever you need it. One sleepy soul at a time. Tonight, you don't need to solve anything. You don't need to process, plan, or figure anything out. The facts you're about to hear will come to you gently. The way understanding sometimes arrives, not through effort, but through simply letting the mind grow quiet and still. Notice the weight of your body right now. Let that weight be held. You don't have to carry anything. Breathe. You are already safe here. Stress begins not in the body, but in a small, almond-shaped region deep in the brain called the amygdala. The moment the amygdala perceives something as threatening, whether a real danger or a worried thought, it sends out a signal that moves faster than conscious awareness. Before you've had a single deliberate thought about a situation, your amygdala has already classified it as safe or unsafe. This is one of the most ancient circuits in the human brain. Shared with mammals who lived hundreds of millions of years ago, the same structure that helped early humans survive predators is the same one now responding to an unanswered email, a deadline, or a conversation that didn't go the way you hoped. It cannot yet tell the difference. That distinction between real threat and imagined one is something only the rest of the brain can slowly teach it. Once the amygdala fires, it activates a cascade that scientists call the hypothalamic pituitary adrenal axis, more commonly known as the HPA axis, the hypothalamus. A small region at the base of the brain signals the pituitary gland, which in turn signals the adrenal glands sitting atop your kidneys. Within moments, these glands release cortisol, the body's primary stress hormone, into the bloodstream. This system is brilliantly designed for acute, short-term threats. Cortisol sharpens attention, mobilizes energy, and prepares the muscles for action. The challenge that researchers have come to understand more deeply over the past 2 decades is what happens when this axis never fully switches off. When the signal keeps arriving day after day, the system that was designed to protect becomes one of the quiet sources of exhaustion that so many people carry without fully understanding why. Cortisol follows a natural rhythm across every 24-hour cycle. In healthy, unstressed individuals, cortisol levels peak sharply in the first 30 minutes after waking. A pattern researchers call the cortisol awakening response. This morning surge is not a sign of stress. It is the body's preparation for the demands of the day ahead. Levels then decline gradually throughout the day, reaching their lowest point in the late evening and early night, which is precisely when the body signals readiness for sleep. Chronic stress disrupts this rhythm. When cortisol remains elevated into the evening hours, the body's descent towards sleep is chemically interrupted. The feeling of lying down exhausted but unable to relax is, in part, a direct consequence of this mismatch. A nervous system still running on daytime chemistry, long after the day has ended. The word stress, as we commonly use it today, was introduced into biology by the Hungarian-Canadian endocrinologist Hans Selye in the 1930s. Selye borrowed the term from physics, where stress describes the force applied to a material. He noticed that laboratory animals exposed to various forms of strain, cold, heat, infection, injury, showed a remarkably similar pattern of biological response, regardless of the specific cause. He called this the general adaptation syndrome, a three-stage process involving alarm, resistance, and exhaustion. What struck Selye most was that the body's response was not unique to any single stressor, but was a universal biological signature of being overwhelmed. His insight changed how medicine understood illness. Many chronic conditions that had previously been categorized separately were recognized, in part, as manifestations of prolonged activation of this same ancient adaptive system. Cortisol is sometimes described as a villain in popular conversation, but the science tells a more nuanced story. Without cortisol, the body cannot function. It regulates blood pressure, support immune response, controls inflammation, and helps convert stored energy into usable fuel. People with Addison's disease, a condition in which the adrenal glands produce insufficient cortisol, experience profound fatigue, dizziness, and can face life-threatening crises during illness or injury because their bodies cannot mount the necessary physiological response. The problem, as with so many biological systems, is not the hormone itself, but the timing and duration of its presence. Cortisol at the right moment, in the right amount, is protective and restorative. Cortisol, sustained at elevated levels across weeks and months, becomes something the tissues and neurons of the brain gradually find difficult to recover from. A landmark study published in the journal Neurology, drawing on data from more than 2,000 adults, most of them in their 40s, found that individuals with the highest blood cortisol levels performed significantly worse on tests of memory, attention, visual perception, and cognitive organization. The connection was particularly pronounced in women. Researchers also found that elevated cortisol was associated with structural changes in the brain, patterns that have historically been associated with early indicators of cognitive decline. What made this study significant was not simply the correlation between stress and cognition, but its scale. It provided some of the strongest population-level evidence that the stress many people carry as a normal feature of midlife is not without measurable consequence on the very organ they rely on to navigate it. The hippocampus, a curved, seahorse-shaped structure buried deep in the temporal lobe, is one of the brain regions most sensitive to sustained cortisol exposure. The hippocampus plays a central role in forming new memories, regulating emotional responses, and, critically, in signaling to the HPA axis that it is safe to stand down. When the hippocampus is functioning well, it acts as a kind of brake on the stress response. But prolonged elevation of cortisol gradually affects the hippocampus's structure and function. Research has shown that chronic stress is associated with a reduction in hippocampal volume over time. This creates a difficult feedback loop. The very structure that would help the body recognize safety and switch off the stress response becomes less able to perform that function, making it harder to return to calm. Sleep and stress exist in a bidirectional relationship that researchers have spent decades mapping. Stress disrupts sleep. This most people know from experience, but the relationship runs in the other direction with equal force. Sleep deprivation activates the stress response. A single night of insufficient sleep has been shown to elevate cortisol levels the following evening. A study from the University of California, Berkeley found that just one night of partial sleep loss was associated with up to a 30% increase in anxiety levels the following day. The mechanism involves both the HPA axis and the amygdala, which becomes more reactive when sleep is reduced. In other words, the nights when falling asleep feels hardest, the nights when the mind is most active with worry, are also the nights when the brain is least neurologically prepared to regulate that worry. The preoptic area of the hypothalamus is the brain's primary sleep regulatory hub, a region that orchestrates the transition from wakefulness into non-rapid eye movement sleep. Research published in Current Biology in late 2023 identified a population of neurons in this region that fire rhythmically during NREM sleep, helping to maintain the continuity of sleep stages. The same study found that stress activates these neurons out of sequence, producing what researchers call microarousals and brief, often unremembered moments of waking that fragment sleep without the sleeper necessarily becoming fully conscious. This means that stress-related sleep disruption is not only about difficulty falling asleep, it also means that even hours logged in bed may not deliver the depth of restoration the body requires, because the architecture of sleep itself is quietly being interrupted throughout the night. Adrenaline, also called epinephrine, is the faster-acting companion to cortisol in the stress response. Where cortisol moves through the bloodstream over minutes and hours, adrenaline acts within seconds. It accelerates heart rate, dilates the pupils, redirects blood flow from digestive organs toward muscles, and heightens sensory alertness. In a genuine emergency, this rapid mobilization is life-saving. In the context of modern chronic stress, it becomes part of a pattern that the body repeats dozens of times a day in response to minor provocations. A loud notification, a tense conversation, a moment of traffic. Each of these triggers a small adrenaline response. Over time, the cumulative effect on the cardiovascular system, digestive system, and immune function is measurable. The body was designed to activate this system rarely and powerfully. It was not designed to sustain low-level activation continuously. The prefrontal cortex, the brain's center of reasoning, planning, and emotional regulation, has a complex and important relationship with the stress response. Under acute stress, blood flow is partially redirected away from the prefrontal cortex toward more reactive, survival-oriented brain regions. This is adaptive in a genuine emergency. You need reflexes more than deliberation when escaping danger. But under chronic stress, this shift becomes more persistent. The prefrontal cortex's ability to modulate the amygdala, to apply context, reason, and perspective to emotional signals, is gradually diminished. This neurological dynamic helps explain why, Under sustained stress, small things feel overwhelming. Minor setbacks feel disproportionately upsetting. And the mental effort required to simply reframe a situation feels exhausting rather than natural. The capacity for equanimity is not a moral quality. It is, in meaningful part, a neurological one. The body does not distinguish between psychological stress and physical stress in terms of the cortisol response it produces. Research comparing individuals under social evaluation stress, being watched and judged, with those undergoing mild physical challenge, found that both produced comparable cortisol elevations. This equivalence has important implications. The social and emotional landscape of daily life, feeling evaluated, uncertain, unacknowledged, or overwhelmed, activates the same biological pathway as more tangible forms of physical hardship. This insight, which has deepened over decades of stress research, helps explain why psychological safety is not a luxury. The feeling of being understood, supported, and free from constant evaluation is not merely pleasant. It is physiologically protective. It is, at the level of hormones and neural circuits, a form of rest. Chronic stress has measurable effects on the immune system, and the relationship is more nuanced than simply weakening immunity. In the short term, acute stress can actually enhance certain immune functions, an adaptive response preparing the body for potential injury. But sustained stress disrupts this regulation. Research has shown that prolonged cortisol elevation suppresses the activity of natural killer cells, reduces the body's ability to mount effective responses to vaccines, and increases levels of inflammatory markers in the blood. Paradoxically, chronic stress is associated with both immunosuppression and chronic inflammation, two states that seem contradictory but can coexist when the regulatory systems become dysregulated. This inflammatory component of chronic stress has become an area of intense research interest, as it appears to connect sustained psychological stress to a range of physical health conditions over time. The gut and the brain are in constant two-way communication through a network of nerves, hormones, and signaling molecules collectively known as the gut-brain axis. The vagus nerve, the longest cranial nerve in the body, carries information not only from the brain down to the digestive system, but upward from the gut to the brain. Approximately 90% of the signals traveling along the vagus nerve travel upward. The gut contains more than 100 million neurons, more than the spinal cord, and produces about 90% of the body's serotonin. Stress disrupts this communication pathway significantly. Changes in gut motility, microbiome composition, and intestinal permeability under chronic stress conditions have been documented. The uncomfortable digestive experiences that so often accompany stressful periods are not imagined. They are a direct expression of this neural and chemical dialogue being thrown off balance. Research published in Nature in 2022 identified a subpopulation of neurons in the ventral tegmental area of the mouse brain, a region involved in both reward processing and stress responses, that activate in response to social defeat, stress, and induce a state of restorative sleep. This finding suggested that the brain may possess a dedicated circuit for initiating sleep specifically in response to psychological stress, a kind of built-in recovery mechanism. While this research was conducted in animal models and requires careful interpretation before applying to human neurobiology, it opens a meaningful scientific question. What if the impulse to sleep after a difficult day is not mere escapism, but a neurologically programmed recovery response that the body, under certain conditions, may be trying to heal itself through the very sleep it seems hardest to reach? One of the most consistent findings in sleep and stress research is that deep sleep, specifically the slow-wave NREM sleep that dominates the early part of the night, functions as a natural regulator of the stress response. A study from the University of California, Berkeley, led by neuroscientist Matthew Walker and postdoctoral researcher Etty Ben-Simon, found that a full night of slow-wave sleep was associated with significant reductions in anxiety the following morning. Brain imaging showed that after sleep, the prefrontal cortex had restored its regulatory capacity. The researchers described deep sleep as a natural anxiolytic, using the same term applied to pharmaceutical anti-anxiety interventions. The implication is both scientifically precise and quietly reassuring. The body already contains, every single night, a mechanism for softening what the day made hard. Telomeres are the protective caps at the ends of chromosomes, often compared to the plastic tips on shoelaces, that help preserve genetic material during cell division. Each time a cell divides, telomeres shorten slightly. When they become too short, the cell can no longer divide and either becomes dormant or dies. Telomere length is now considered one of the biological markers of cellular aging. Sustained psychological stress has been associated with accelerated telomere shortening in multiple peer-reviewed studies. Research on caregivers of chronically ill family members found significantly shorter telomere lengths compared to non-caregivers of the same age. The finding suggests that the experience of prolonged psychological stress does not merely feel aging at the level of chromosomes. In certain measured ways, it may accelerate the biological processes associated with it. The body's stress response evolved in a world in which stressors were typically brief and physically resolved. A predator encountered, a threat escaped, a resource secured. The physiological aftermath of the response was therefore designed to be short. The problem that modern neuroscience has helped clarify is that the human capacity for abstract thought, for anticipating future threats, replaying past ones, and sustaining worry about situations that are not immediately present, means the stress response can be activated by things that exist only as thoughts. A lion that has left can no longer harm you. A worry about tomorrow has not yet happened. But the amygdala and the HPA axis respond to both with similar chemistry. The mind's ability to simulate the future, one of its most extraordinary capabilities, is also one of the primary reasons the stress response so rarely gets to fully switch off. Allostatic load is a term coined by neuroscientist Bruce McEwen to describe the cumulative wear and tear on the body that results from chronic stress and the adaptive responses it demands. Unlike the immediate stress response, allostatic load accumulates invisibly over time. In the slow degradation of regulatory systems, the gradual shifts in baseline hormone levels, the incremental effects on cardiovascular, immune, and neural tissues. McEwen's research demonstrated that the body's systems are not simply restored to baseline after each stress episode. Under repeated or sustained stress, the baseline itself shifts. What the body considers normal becomes recalibrated around a state of higher alert. This recalibration is not experienced as dramatic change, but as a quiet, persistent heaviness, a sense of never quite recovering, never quite feeling as rested as the sleep seemed to promise. Research across multiple populations has found that women are more likely than men to report psychological symptoms of stress, including anxiety, difficulty sleeping, and emotional rumination, while men are more likely to report behavioral symptoms such as increased substance use or social withdrawal. These differences are not simply cultural. They have neurobiological correlates. Estrogen appears to sensitize the stress response in certain contexts, while also playing protective roles in others. The hippocampus and amygdala contain estrogen receptors and hormonal fluctuations across the menstrual cycle, perimenopause, and menopause influence how the stress response is regulated. This means that the stress experience is not uniform across individuals. It is shaped by biology, history, and the specific hormonal environment of the nervous system at any given period of life. The term rumination in psychology describes the tendency to repeatedly return to negative thoughts, particularly about past events or future concerns, without moving toward resolution. Rumination is one of the most reliably identified contributors to both chronic stress and sleep difficulty. It is distinct from reflection, which involves examining an experience with the aim of understanding or learning from it. Rumination is cyclical. It returns to the same ground without progressing. Research using functional brain imaging has found that ruminative thinking is associated with heightened activity in the default mode network, the set of brain regions most active during self-referential thought and mind-wandering. When the default mode network is highly active at bedtime, the transition into sleep is significantly disrupted. The mind, in a sense, cannot easily leave the conversation it keeps having with itself. The social environment is among the most potent modulators of the stress response. Research on social support and stress physiology consistently shows that the presence of a trusted individual during a stressful task significantly reduces cortisol output compared to facing the same task alone. Conversely, social isolation and the chronic experience of loneliness activate the same HPA axis pathways as other forms of stress. Studies have found that lonely individuals show elevated cortisol levels, more fragmented sleep, and increased activity in threat detection brain regions. The mechanism involves not just psychology but neurochemistry. Oxytocin, the neuropeptide associated with social bonding, has direct inhibitory effects on the amygdala and the stress axis. Connection, at the level of brain chemistry, is genuinely calming. Its absence is genuinely stressful. In the most physiological sense of that word, breathing is one of the few autonomic functions, processes that operate without conscious control, that can also be deliberately regulated. This unique quality makes it a direct access point to the autonomic nervous system. Slow, deep diaphragmatic breathing activates the parasympathetic nervous system, the branch responsible for rest, digestion, and recovery, and has been shown to reduce cortisol levels, lower heart rate, and diminish activity in the amygdala. Research on practices that emphasize controlled breathing, including certain forms of meditation and yoga, consistently demonstrates measurable reductions in stress biomarkers. The physiological mechanism involves the vagus nerve. Slow exhalations stimulate vagal tone, increasing the heart rate variability that is itself a marker of nervous system resilience. Each slow, deliberate breath is not merely calming in feeling, it is calming in chemistry, in neurology, and in the measurable state of the stress response. Sleep architecture. The precise cycling between different stages of sleep across the night is significantly altered by chronic stress. NREM sleep is divided into 3 stages, with stage 3 slow-wave sleep representing the deepest and most physiologically restorative phase. Research has found that individuals reporting high chronic stress show reductions in the proportion of time spent in stage 3 sleep and increases in the lighter stage 1 and stage 2 sleep. REM sleep. The stage associated with emotional memory processing and the integration of difficult experiences is also disrupted by elevated cortisol in the evening hours. Since cortisol suppresses REM onset, this means that the 2 sleep stages most associated with emotional recovery and physical restoration are precisely the ones that stress most reliably erodes. The stress response, for all its costs, is not a design flaw. It is an ancient, extraordinarily effective survival system that has protected our species across vast stretches of evolutionary time. The organisms that did not have a robust stress response did not survive long enough to pass their genes forward. This perspective matters not as a justification for the suffering that chronic stress causes, but as a reframing of the relationship between the body and its own responses. The nervous system that keeps you tense when you want to be still, that races when you want quiet, is not broken. It is doing precisely what it was built to do: scanning, protecting, preparing. Understanding this is the first step in the question this video follows. Can understanding stress science actually help your brain let go of it? The answer the science suggests, quietly and consistently, is yes. For just a moment, let the facts rest. Notice your body right now, the places where it still holds tension, perhaps your shoulders, slightly raised, perhaps your jaw, not quite released, perhaps a tightness behind the eyes. You don't need to force anything open. Just notice. Recognition itself is a form of softening. Your nervous system has been listening to everything you've heard tonight, and somewhere beneath the thinking, something in you already understands. The stress was never the enemy. It was trying to protect you. For now, let it know it can rest. Let your body know the same. Breathe in slowly and let go, just a little more than before. The autonomic nervous system has 2 primary branches: the sympathetic system, which activates the stress response and prepares the body for action, and the parasympathetic system, which governs rest, digestion, and recovery. These 2 branches are not simply on and off switches. They exist in dynamic tension, with their relative balance determining the body's physiological state at any given moment. Heart rate variability. The natural variation in time between heartbeats is one of the best measurable indicators of this balance. High heart rate variability indicates a flexible, responsive nervous system with strong parasympathetic tone. Chronic stress tends to reduce heart rate variability over time, reflecting a nervous system that has gradually lost some of its capacity to move fluidly between alertness and recovery. Neuroplasticity. The brain's capacity to reorganize itself by forming new neural connections is influenced by the stress environment. Moderate, manageable stress appears to support certain forms of neuroplasticity, particularly in regions involved in learning and adaptation. But chronic, uncontrollable stress has the opposite effect on key regions. Research has shown that sustained stress reduces the density of dendritic spines, the tiny protrusions on neurons that receive incoming signals, in the prefrontal cortex and hippocampus, while increasing them in the amygdala. In functional terms, this means that chronic stress simultaneously makes the brain's threat detection center more elaborately wired, while reducing the complexity of connections in the areas responsible for contextual reasoning and emotional regulation. The architecture of the stressed brain literally shifts toward reactivity. Cortisol has a direct suppressive effect on REM sleep. Because REM sleep is the stage during which the brain most actively processes emotional memories, consolidating them, reducing their emotional charge, and integrating them into a broader narrative of experience, this suppression has meaningful consequences. Matthew Walker's research group has described REM sleep as a form of overnight therapy during which the brain revisits difficult experiences without the stress neurochemistry that accompanied them originally. When cortisol prevents or fragments REM sleep, this processing is incomplete. Emotionally charged memories retain more of their raw intensity. The day's stresses arrive into the next morning, less metabolized, less softened. Sleep, in this sense, is not an escape from difficulty, but the biological process through which difficulty is gradually transformed. The perception of control, or the lack of it, is one of the most significant factors determining whether a stressor activates the HPA axis in a sustained way. Research consistently shows that exposure to uncontrollable stressors produces greater and more prolonged cortisol responses than exposure to equally demanding stressors that allow for some form of agency. Studies on learned helplessness, a state in which repeated exposure to unavoidable stress leads to passivity even when escape becomes possible, show distinct neurobiological signatures, including reduced prefrontal cortex activity and altered stress hormone regulation. The experience of helplessness is not merely psychological, it reshapes the brain's stress regulatory circuitry. Conversely, even small, symbolic acts of agency during stressful periods appear to partially buffer the physiological impact of the stress itself. The phenomenon known as stress contagion describes the measurable transfer of stress responses between individuals in close proximity. Research published in Psychoneuroendocrinology found that observing another person in a stressful situation, even through a one-way mirror or via video, was sufficient to elevate cortisol levels in approximately 26% of observers. When the person being observed was a close relationship partner, this figure rose significantly. The mechanism involves both the mirror neuron system and direct hormonal pathways. This finding has practical implications for sleep. The stress levels of those we share our close environment with are not separate from our own physiology. The tense household, the worried partner, the anxious household energy that lingers into evening, these are not merely emotional impressions. They have measurable biological correlates in the people who inhabit them. PT-INF, brain-derived neurotrophic factor, is sometimes described as fertilizer for the brain. It supports the growth, maintenance, and survival of neurons, and is essential for the synaptic plasticity that underlies learning and memory. Chronic stress reliably suppresses BDNF expression, particularly in the hippocampus. This suppression is thought to be one of the mechanisms connecting chronic stress to both cognitive changes and increased vulnerability to depression. Physical exercise is one of the most reliably documented ways to increase BDNF levels, a finding that helps explain why regular movement provides measurable cognitive and emotional protection even under sustained stress. Sleep also restores BDNF levels after stress-induced suppression, adding another dimension to the relationship between rest and neural recovery. Research on post-traumatic stress disorder has illuminated the extremes of how the stress response can become dysregulated. In PTSD, the normal recovery arc of the stress response, activation, resolution, restoration, becomes interrupted. The amygdala remains in a state of heightened reactivity. The prefrontal cortex's regulatory influence is diminished, and the hippocampus's ability to properly contextualize and date memories is impaired. Traumatic memories are experienced not as past events, but as present ones, stripped of temporal context by hippocampal disruption. While PTSD represents the far end of stress dysregulation, the neurobiological mechanisms it reveals are continuous with ordinary stress physiology. Understanding PTSD helped researchers understand, in sharper detail, what more ordinary forms of sustained stress do to the same brain regions over time. The microbiome, the community of trillions of microorganisms living in the digestive tract, is now understood to communicate bidirectionally with the brain through what researchers call the gut-brain axis. Stress alters the composition of the gut microbiome in measurable ways. It reduces microbial diversity, decreases populations of certain beneficial bacteria, and increases intestinal permeability. Conversely, certain microbiome compositions appear to buffer stress reactivity. Research published in Nature Microbiology has found associations between gut microbiome diversity and both cortisol reactivity and anxiety levels. The precise mechanisms remain an area of active investigation, but the finding that the bacteria in the digestive tract may influence how the brain responds to stress has expanded the biological understanding of the stress response well beyond the brain and nervous system, where it was once thought to exclusively reside. Inflammation is now understood to be both a consequence and a contributor to chronic stress. Elevated cortisol over the long term paradoxically contributes to a state of low-grade systemic inflammation, partly because the immune system's sensitivity to cortisol's anti-inflammatory signals decreases with prolonged exposure, a process called glucocorticoid resistance. This low-grade inflammation has been linked to disrupted sleep, depressed mood, and cognitive changes. Research by psychologist Sheldon Cohen at Carnegie Mellon University found that individuals with chronically stressful experiences showed elevated inflammatory markers, and that these markers mediated some of the health effects associated with stress. The implication is that what we experience as psychological stress leaves a measurable inflammatory signature in the blood, a biological record of what the mind has been carrying. Music has documented effects on the physiological stress response. Research published in PLOS ONE found that listening to slow, self-selected music before a stressful task significantly reduced cortisol levels and subjective anxiety compared to sitting in silence. The mechanism involves multiple pathways. The auditory cortex connects directly to the limbic system. Music modulates autonomic nervous system tone, and familiar music activates reward circuitry that releases dopamine. Slow music, with tempos between 60 and 80 beats per minute, close to a resting heart rate, appears particularly effective at promoting parasympathetic activation. This science helps explain why ambient sound, gentle music, or simply the absence of stimulating input before sleep is not merely a preference. It is a physiologically meaningful part of preparing the nervous system for rest. The concept of eustress, a term also coined by Hans Selye, describes stress that is experienced as positive, motivating, and within the individual's sense of capacity to manage, presenting to an audience you care about, completing a challenging physical feat, or navigating a meaningful creative problem can all produce measurable stress responses while being experienced subjectively as energizing rather than depleting. Research has found that the cognitive appraisal of a stressor, whether one frames it as a threat or as a challenge, influences the specific hormonal and cardiovascular profile of the stress response, even when the objective demands are identical. The distinction is not merely semantic. Challenge appraisals produce stress responses with a more favorable cardiovascular pattern, while threat appraisals produce more cortisol and greater vasoconstriction. Cortisol's suppression of the immune response has a nuanced relationship with sleep. Research has found that during slow-wave sleep, the balance of the immune endocrine system shifts toward immune-enhancing activity. T-cell production increases, and certain cytokines associated with immune memory are released. Growth hormone, which plays roles in tissue repair and immune modulation, is also secreted primarily during deep NREM sleep. This means that the restorative function of sleep is not limited to neural recovery. It is also a window in which the immune system consolidates its defenses. Chronic stress, by eroding deep sleep, therefore impairs not only mood and cognition, but also the nightly maintenance of immune function that sleep is designed, in part, to provide. The perception of time changes measurably under stress. Research has found that individuals in high-stress states tend to overestimate how much time has passed and also how much time demanding tasks will take. This temporal distortion is partly mediated by the heightened attentional processing that the stress response induces. More information is processed per unit of time, so each moment feels denser, fuller, and longer. For sleep, this distortion is particularly relevant. The wakeful hours of a stressed night often feel far longer than they are. The time spent lying awake in worry genuinely is, in some neurological sense, experienced as longer, not just perceived that way. This understanding does not reduce the discomfort, but it offers a small correction to the narrative. The night is not necessarily as endless as it feels. Mindfulness-based stress reduction, known as MBSR, is an 8-week program developed by John Kabat-Zinn at the University of Massachusetts in the late 1970s that has since been studied in hundreds of controlled trials. The core practice involves deliberately directing sustained, non-judgmental attention to present moment experience, including breath, body sensation, and the arising and passing of thoughts and emotions. Meta-analyses of MBSR research have found consistent reductions in self-reported stress, anxiety, and depression, as well as measurable changes in cortisol levels and inflammatory markers. Brain imaging studies have documented changes in the prefrontal cortex, hippocampus, and amygdala following MBSR training. Structural and functional shifts that align with reduced stress reactivity. The practice does not eliminate stress. It changes the brain's relationship to it. The default mode network is a set of brain regions, including the medial prefrontal cortex, posterior cingulate cortex, and lateral parietal cortex, that become most active when the mind is not engaged with a specific external task. It is the network of self-referential thought, autobiographical memory, and mental simulation of past and future. In individuals with high chronic stress, the default mode network tends to show hyperactivity during rest. The mind, even when given space, continues generating stress-related content. This is the neurological basis of the experience many describe as the inability to simply sit quietly. The brain's resting state has been recruited into the stress response, training the mind to rest. Through practices that quiet the default mode network is, neurologically, a form of stress intervention. You've been listening for a while now. Perhaps your breathing has slowed. Perhaps the weight of the day feels slightly further away. This channel exists to be here on nights like this one. A gentle like or subscribe helps more people find their way here too. That's all. Back to the science. The relationship between exercise and the stress response is one of the most consistently documented in psychobiological research. Moderate aerobic exercise produces an acute cortisol response during activity. The body treats exertion as a form of managed stress, but this is followed by a significant reduction in cortisol levels in the recovery period. Regular exercise lowers baseline cortisol levels over time, improves heart rate variability, increases BDNF expression, and promotes deeper NREM sleep. Research suggests that even a single session of moderate exercise, a 30-minute walk at a brisk pace, can reduce cortisol levels for several hours afterward and improve sleep quality that night. The stress buffering effect of movement is one of the most accessible and well-evidenced tools available, requiring no equipment beyond the body itself. Social laughter. Genuine laughter shared with others, rather than solitary or performed laughter, has measurable effects on the stress axis. Research has found that anticipating a humorous experience is itself sufficient to reduce cortisol and adrenaline levels. Genuine laughter activates the parasympathetic nervous system, increases endorphin release, and has been associated with reductions in inflammatory markers. Laughter also modulates the default mode network, interrupting ruminative loops in ways that share some mechanistic similarity with mindfulness practice. These findings are not trivial. They suggest that the social and playful dimensions of human experience, the sharing of humor, the ease of genuine connection, are not simply pleasant adjuncts to a serious life. They are physiologically meaningful contributors to stress regulation. The stress response has a measurable effect on pain perception. Under acute stress, the body temporarily elevates pain thresholds, a phenomenon called stress-induced analgesia, mediated partly by the release of endogenous opioids. This is why injuries sustained during high adrenaline moments are often not fully felt until the adrenaline subsides. Under chronic stress, however, the relationship inverts. Persistent HPA axis activation increases sensitivity to pain over time, partly through its effects on the central nervous system's pain-modulating circuits and partly through the inflammatory pathways that chronic cortisol elevation promotes. The chronic stress that makes sleep harder also, in many individuals, makes the body feel more uncomfortable at rest. Another layer of the feedback loop that keeps the nervous system from fully settling. Nature exposure, time spent in natural environments, has a well-documented stress buffering effect. Research using cortisol measurements before and after time in forests, parks, and other natural settings consistently finds reductions in stress hormone levels following nature exposure. Japanese researchers investigating the practice of Shinrin-yoku, forest bathing, have documented reduced cortisol, lower blood pressure, decreased sympathetic nervous activity, and enhanced parasympathetic tone following time in forested environments compared to urban environments. The mechanisms include visual complexity of a specific kind, natural fractals that appear to reduce frontal lobe effort, as well as phytoncides, the aromatic compounds released by trees, which have documented immune modulating and cortisol-reducing effects when inhaled. The body, it appears, recognizes something restoring in the presence of living systems. Memory consolidation, the process by which new information is stabilized into long-term memory, occurs primarily during sleep, particularly during slow-wave NREM and REM stages. Stress affects memory consolidation in complex, bidirectional ways. The stress hormones released during learning can actually enhance the encoding of emotionally significant memories, explaining why stressful or emotionally charged events are often remembered with particular vividness. But stress in the consolidation window, the period of sleep following learning, impairs the integration of neutral and procedural memories. The memory system under stress becomes selective. It prioritizes threat-relevant information, consolidating fear and worry while allowing more neutral or positive information to fade. The stressed brain is not simply a less effective memory system, it is a differently prioritized one. Chronic stress is associated with changes in the structure and function of the prefrontal cortex that extend beyond functional suppression during acute stress episodes. Long-term imaging studies have found that individuals reporting higher chronic stress levels show reduced gray matter density in the prefrontal cortex. This structural change parallels the functional changes: reduced capacity for top-down regulation of the amygdala, less flexibility in emotional response, and greater difficulty with tasks requiring cognitive control. These structural changes are not permanent. The brain retains neuroplasticity across the lifespan, and the reversal of chronic stress conditions is associated with partial recovery of prefrontal structure and function. But the changes are also not trivial. They represent a physical record of what sustained stress asks of the brain over time. Perceived stress is often more predictive of physiological outcomes than objective measures of life difficulty. The Perceived Stress Scale, developed by Sheldon Cohen in 1983, asks individuals to assess how overwhelmed, uncontrollable, and unpredictable their lives feel. not what has objectively happened. Studies using this scale have found that perceived stress scores predict cortisol profiles, immune function, sleep quality, and cardiovascular health more reliably than external assessments of life stressors. This finding underscores something that stress researchers have consistently emphasized. The stress experience is not simply a reflection of events. It is a product of events filtered through personal history, cognitive style, available resources, and the specific neurobiological setpoint of the individual. The same situation can produce very different stress responses in different people. And this is not weakness. It is complexity. The relationship between stress and food intake is mediated by multiple hormonal and neural pathways. Cortisol stimulates appetite, particularly for foods high in sugar and fat, by influencing appetite-regulating hormones, including ghrelin and leptin. This is thought to be an adaptive response. Under acute stress, calorie-dense food provides rapid energy for physical action. But under chronic stress, where action is rarely taken and cortisol remains chronically elevated, this appetite stimulation contributes to dysregulated eating patterns. The foods that stress most powerfully calls for, highly processed, high sugar, high-fat foods, also tend to promote inflammatory responses and disrupt gut microbiome composition. The dietary patterns associated with chronic stress therefore feed back into the inflammatory and microbiome pathways that themselves sustain the stress response. Writing, specifically expressive writing about stressful or emotionally significant experiences, has been studied as a stress intervention by psychologist James Pennebaker at the University of Texas since the 1980s. Across dozens of controlled trials, writing about difficult emotional experiences for 15 to 20 minutes on 3 to 4 occasions has been associated with reductions in stress symptoms, improvements in immune function, fewer medical visits, and better sleep quality. The mechanism appears to involve the translation of emotionally fragmented experience into coherent narrative, a process that activates the prefrontal cortex and reduces the amygdala's continued reactivity to unresolved emotional content. Naming and organizing experience, it seems, does something to its neurological weight. The act of putting things into words is not merely cathartic. It is, in measurable ways, metabolizing. The capacity for resilience, the ability to adapt to adversity without sustained dysregulation of the stress response, is shaped by both genetic factors and life experience, and is also modifiable. Research on resilience identifies several consistent neurobiological correlates: strong prefrontal cortex amygdala connectivity, good regulation of the HPA axis, healthy social support networks, and adequate sleep. Crucially, resilience is not the absence of stress reactivity. Resilient individuals still show cortisol responses to stressors. What distinguishes them is the speed and completeness of their recovery. The stress response activates, does its job, and then subsides. This recovery capacity, the return to baseline, is, in many ways, more central to stress health than the magnitude of the initial response itself. You are halfway through tonight's journey. Take a moment to feel what has shifted, perhaps not dramatically, but in small ways. Perhaps the breath has found its own rhythm. Perhaps the body has grown heavier against whatever holds it. you've been learning about stress, about what it does in the body, how it speaks through the nervous system, why it sometimes refuses to let go. And perhaps something in hearing this has made it feel slightly less mysterious, slightly less like something happening to you, and slightly more like something you can begin to understand. Rest in that understanding. Let it be enough for now. You are exactly where you need to be. The stress response evolved in a world without artificial light. Light, particularly blue spectrum light, signals the suprachiasmatic nucleus in the hypothalamus to suppress melatonin production. Keeping the body alert, stress and light exposure interact in important ways. A stressed nervous system is already primed for vigilance, and exposure to screen light in the hours before bed amplifies this alertness by adding a second signal, instructing the body to remain awake. The combination of elevated cortisol and light-induced melatonin suppression creates conditions in which the biological preparation for sleep is doubly undermined. Not by a single cause, but by two reinforcing systems working against rest simultaneously. Research on sleep and stress has identified a pattern called pre-sleep cognitive arousal, the tendency for the mind to become more, not less, active in the period just before sleep. For individuals with high chronic stress, this arousal involves intrusive thoughts, worry cycles, and mental rehearsal of unresolved problems. Studies measuring brain activity during pre-sleep wakefulness in high-stress individuals find elevated activity in regions associated with working memory and self-referential thought. The experience is familiar. The mind, denied the distractions of the day, turns to whatever is unresolved. Understanding this pattern does not eliminate it, but it situates it, transforms it from evidence of personal failure into a well-documented neurological phenomenon shared by many. The adrenal glands, two small triangular structures sitting atop the kidneys, weigh only about 10 grams each. Yet the hormones they produce influence virtually every system in the body: cardiovascular, immune, metabolic, reproductive, and neurological. Under chronic stress, the adrenal glands are asked to sustain output of cortisol and adrenaline at levels that were designed to be temporary. Research has documented changes in adrenal gland volume and function in individuals with sustained high-stress exposures. The small, tireless glands that mediate so much of the stress response were built for urgency, not endurance. Their steady, quiet labor under chronic stress is one of the least seen dimensions of what the body carries through difficult seasons of life. Temperature regulation is intertwined with the stress response. Cortisol and adrenaline both influence thermoregulation. Adrenaline causes peripheral vasoconstriction that can produce cold hands and feet even in warm environments. Sleep, conversely, requires a modest drop in core body temperature, a drop of approximately 1 degree Celsius. To initiate properly, the heightened peripheral vasoconstriction associated with stress disrupts this thermal drop, adding a physiological barrier to sleep onset that operates below conscious awareness. The simple act of warming the hands and feet before bed, which promotes heat dispersal from the core, has been shown to accelerate sleep onset, partly by facilitating the body temperature drop that stress-induced vasoconstriction was preventing. Research using ecological momentary assessment The practice of measuring stress and mood multiple times per day through smartphone prompts has found that daily stress levels are not uniformly distributed. They peak in mid-morning and again in late afternoon, corresponding to the natural cortisol rhythm and its interaction with workday demands. Evening measurements consistently show the lowest daily stress levels for most individuals, a finding that suggests the body does have a natural inclination toward evening deceleration. The challenge is that cognitive and digital habits, continuing to check communications, engage with stimulating content, or review tomorrow's demands into the late hours, can override this natural physiological drift toward calm. Chronic stress has measurable effects on reproductive physiology. Sustained activation of the HPA axis suppresses the hypothalamic pituitary gonadal axis, the hormonal pathway regulating reproductive function. In women, this suppression can contribute to irregular menstrual cycles and changes in hormonal balance. In men, elevated cortisol is associated with reduced testosterone levels. These effects are understood as adaptive. From an evolutionary standpoint, prolonged resource scarcity or threat is not an appropriate context for reproduction. The body's stress systems, in essence, communicate to reproductive systems that conditions are not yet safe. The connection between stress and reproductive health is not psychological. It is a direct hormonal dialogue between two of the body's most ancient regulatory systems. Research on secondary traumatic stress, the stress that caregivers, healthcare workers, and first responders experience through sustained exposure to others' suffering, has expanded understanding of how stress is absorbed through empathic engagement. Neuroimaging studies have found that witnessing pain activates some of the same neural circuits involved in experiencing pain directly. For individuals in high empathy, high exposure roles, the cumulative activation of these circuits creates a stress burden that is genuine, measurable, and often unrecognized. The biology of compassion includes a real physiological cost, one that accumulates in the same stress pathways as more direct forms of adversity, and that deserves the same quality of rest and recovery. Positive emotions do not simply represent the absence of stress. They have distinct, active effects on stress physiology. Research by psychologist Barbara Fredrickson at the University of North Carolina has documented what she calls the broaden and build theory. Positive emotional states broaden the attentional field, increase cognitive flexibility, and build psychological and physiological resources over time. At the neuroendocrine level, positive affect is associated with lower baseline cortisol, a faster cortisol recovery following stress exposure, and better sleep quality. Gratitude specifically, the deliberate acknowledgement of what is present and valuable, has been associated in multiple studies with lower pre-sleep cognitive arousal, fewer intrusive thoughts at bedtime, and longer sleep duration. The ancient practice of counting what is good turns out to have a measurable neurochemical correlate. The vagal brake, a concept from polyvagal theory developed by neuroscientist Stephen Porges, describes the ability of the myelinated vagus nerve to rapidly and flexibly modulate heart rate and autonomic state. When the vagal brake is functioning well, the nervous system can shift smoothly between states of social engagement, gentle alertness, and deep rest. Chronic stress weakens this flexibility, reducing vagal tone and making the transitions between states slower and less complete. Research supporting vagal tone as a measurable index of stress resilience has found associations between resting vagal tone, measured through heart rate variability, and emotional regulation capacity, social connection, quality, and recovery speed following stress exposure. Note that polyvagal theory remains an area of ongoing scientific discussion, and its full clinical applications are still being validated. Chronic stress is associated with changes in the density and sensitivity of glucocorticoid receptors, the cellular docking points through which cortisol exerts many of its effects. With prolonged cortisol exposure, cells can down-regulate their receptor density as a form of protection, reducing sensitivity. This receptor resistance means that cortisol's natural anti-inflammatory and regulatory functions become less effective over time, paradoxically resulting in greater inflammation. Even as cortisol levels remain elevated, the immune dysregulation associated with chronic stress is therefore not simply about cortisol being too high. It is about the body's response to cortisol becoming less precise. The regulatory system loses some of the fine-tuning that made it effective. Research using functional magnetic resonance imaging has found that the neural response to negative emotional imagery is significantly dampened after a full night of sleep compared to an equivalent period of wakefulness. The mechanism involves REM sleep's role in reducing the amygdala's reactivity to emotional stimuli while consolidating the contextual information provided by the hippocampus. In practical terms, the experiences and concerns that feel most urgent and most distressing in the late evening are genuinely processed differently, less rawly. After sleep, this is not a platitude. It is a documented neurological phenomenon. The brain that wakes from sufficient sleep has literally altered its emotional response circuitry during the hours of rest. The concept of stress inoculation, deliberate, graduated exposure to manageable challenge, has been developed both as a therapeutic approach and as a training paradigm for high-stress professions. The principle is that controlled exposure to stress, with adequate recovery, strengthens the regulatory systems that buffer stress. Research on stress inoculation training finds improvements in cortisol response profiles, smaller, better bounded stress responses, and faster return to baseline. Following graduated exposure programs, the physiological analogy to vaccination is meaningful. A small, managed challenge that teaches the regulatory system what it is capable of. The capacity to cope is not fixed. It is something that can be built gradually and deliberately over time. Scent has a unique and direct connection to the stress response. The olfactory nerve, which carries smell information from the nose, is the only sensory system with a direct projection to the amygdala and hippocampus without first passing through the thalamic relay that other senses traverse. This anatomical directness means that smell reaches emotional and memory-associated brain regions with unusual speed and intimacy. Research has found that certain scents, lavender in particular, produce measurable reductions in cortisol levels and sympathetic nervous activity. The calming effect of lavender is not merely cultural. It has documented neurochemical correlates. The chemistry of rest is available, in part, through the air the body is already breathing. The hypothalamus, about the size of an almond, is sometimes called the master regulator of homeostasis. The body's drive to maintain internal stability in the face of external change It monitors temperature, taihunga, thirst, circadian rhythms, and the stress response simultaneously, adjusting outputs to maintain equilibrium across all these systems. Chronic stress taxes the hypothalamus by demanding sustained activation of the HPA axis, while also managing the downstream effects of this activation on sleep, appetite, and circadian timing. The hypothalamus is not a system designed for chronic conflict between its own regulatory functions. Its elegant design assumes that stress is temporary and that the rest it orchestrates will have time to do its work. Stress-related sleep disturbances are among the most commonly reported health concerns globally. The American Psychological Association's annual Stress in America survey consistently finds that sleep is among the top activities Americans report doing less of when stressed, and simultaneously report stress as among the most common causes of their sleep difficulties. This circular relationship, stress disrupting sleep, sleep deprivation worsening stress, is described by researchers as a bidirectional maintenance cycle. Breaking this cycle typically requires addressing both sides simultaneously. Interventions that target only sleep without addressing stress, or only stress without addressing sleep, tend to produce more limited results than approaches that engage both dimensions of the cycle at once. Writing and narrative. The translation of experience into story appears to have a distinct neurological function in stress processing. Research suggests that when emotionally difficult experiences remain in a non-narrative form, as fragmented sensory impressions, emotional states, or unresolved tensions, they continue to activate stress-related neural circuits. When they are translated into coherent narrative, the prefrontal cortex is engaged in a way that reduces the amygdala's sustained reactivity. This is why therapy modalities that emphasize narrative construction, telling the story of what happened and what it meant, often produce reductions in stress physiology alongside psychological insight. The brain, it seems, is partly calmed by coherence, by the quiet act of making sense. Research on sleep in older adults has found that the architecture of sleep changes with age in ways that parallel some of the effects of chronic stress. Slow-wave NREM sleep decreases with age. REM sleep becomes less consistent, and sleep becomes more fragmented. Some researchers have proposed that this age-related sleep change shares neurobiological pathways with the stress-related sleep changes documented in younger adults. The possibility that years of accumulated stress leave a lasting imprint on the structure of sleep beyond what aging alone would produce is an area of ongoing research. What is clear is that the sleep system, like the stress response system, accumulates history. The past is present in the night, in ways both visible and not yet fully understood. The body's circadian clock, the 24-hour biological rhythm orchestrated by the suprachiasmatic nucleus and expressed through the body's tissues, regulates not only sleep and wakefulness, but also the timing of cortisol release, immune activity, metabolism, and cellular repair. Chronic stress disrupts circadian rhythms. Elevated evening cortisol delays the body's transition into the physiological state associated with sleep. And fragmented sleep then feeds back to destabilize the circadian signal. Research has found that circadian disruption is itself a physiological stressor. The misalignment between the body's internal clock and external behavioral patterns produces its own elevation of stress hormones. In this sense, restoring sleep regularity, consistent timing of sleep and waking, is also, in a physiological sense, a stress intervention. Research on compassion-focused therapy has found that developing an attitude of self-compassion, treating oneself with the same kindness one would offer a friend in difficulty, produces measurable reductions in cortisol reactivity to stress. Studies by Kristin Neff at the University of Texas and others have found that higher self-compassion scores are associated with lower chronic stress levels, better sleep quality, a more rapid cortisol recovery following stress exposure. The mechanism appears to involve reduced threat-based self-evaluation. When the inner critic quiets, the threat response it was, generating quiets with it. Self-compassion is not a softening of standards. It is a neurological adjustment of the self-monitoring system that stress so often turns against the person it inhabits. The relationship between stress and creativity is complex and phase-dependent. Acute stress can temporarily sharpen focused attention and narrow cognitive scope in ways that impair divergent, associative thinking, the mode most associated with creative insight. But moderate stress, held within manageable range, can enhance motivation and task engagement. Sleep, and particularly REM sleep, is strongly associated with creative insight. The loosely associative processing of REM creates connections between disparate elements of memory and experience that waking, focused thought often does not. Several well-documented creative breakthroughs in science and art have been attributed to solutions that arrived during sleep or in the hypnagogic state just before it. Rest, in this sense, is not the opposite of productive thought. It is often where productive thought completes itself. Research using wearable technology to monitor physiological stress indicators, heart rate variability, skin conductance, and movement patterns across entire days has revealed the accumulation of micro-stressors, small, individually insignificant stressors that add up across the day into a measurable physiological burden. These include brief frustrations, minor interpersonal frictions, small uncertainties, and moments of cognitive overload. Each is too small to notice consciously. Their aggregate, however, is physiologically significant. This research helps explain why many people arrive at the end of a day that contain no single major event feeling profoundly depleted, carrying the weight of dozens of small activations of the stress system that were each beneath the threshold of conscious attention, but none beneath the threshold of physiological response. Nature's fractal patterns, self-similar geometric structures that repeat at different scales, found in coastlines, fern leaves, river branches, and clouds, have been found to reduce stress in ways that are measurable through both cortisol levels and alpha brainwave activity. Research by physicist Richard Taylor at the University of Oregon found that viewing fractal patterns with a specific complexity range, mid-range fractal dimension, found commonly in natural environments, produced reductions in physiological stress indicators of up to 60%. The visual system appears to process natural fractals with unusual efficiency, reducing the neural effort required for scene interpretation and allowing attention to soften and rest. The natural world, at its structural level, may be formatted for recovery. The anticipation of stress, worrying about a future event, activates many of the same neurobiological pathways as experiencing the stress directly. Research using cortisol measurement has found that individuals told they will undergo a stressful evaluation show cortisol increases beginning hours before the event, continuing through the event, and persisting afterward. For those with high trait anxiety, this anticipatory stress response is more pronounced and longer lasting. This means that the total biological cost of a stressful event is substantially greater than the event itself. It includes the extended period of anticipatory activation beforehand and the rumination that follows after. Worry is not merely a psychological experience. It is a physiological one, with a biochemistry that the body bears across time. Research on the long-term effects of childhood adversity, conducted through the framework of adverse childhood experiences, known as ACEs, has documented how early stress exposure shapes the stress response system across a lifetime. Children who experience high levels of adversity show altered HPA axis calibration, changes in hippocampal development, and different amygdala reactivity profiles compared to children in lower stress environments. These changes are not merely psychological. They are reflected in epigenetic patterns, stress hormone profiles, and sleep architecture into adulthood. The stress system calibrates itself to the environment it grows up in. Understanding this is not an invitation to resignation. It is an invitation to the specific compassion due to a nervous system that learned what it learned in order to survive. The science of stress returns, again and again, to a finding that is quietly remarkable. The body wants to recover, given adequate conditions, sufficient sleep, social connection, manageable demands, and the simple biological opportunity to rest. The stress response system does not remain elevated indefinitely. It returns to baseline. Even after prolonged activation, the regulatory systems retain plasticity. This is the answer, the question this video has been building toward. Can understanding stress science actually help your brain let go of it? What the science suggests is yes. That understanding creates the very conditions for recovery through the accumulated effect of recognizing what the body needs, and where possible, providing it. When science names what your body has quietly carried, the nervous system finally has permission to rest. Everything that was activated can return to stillness. This is not hope. This is biology. The same system that raised your cortisol this morning has pathways built in, always present, for bringing it back down. The same nervous system that learned vigilance can also learn rest. Tonight is part of that learning, not a dramatic shift, just one quiet evening in which the body is given what it is designed to receive. Let your muscles soften. Let your breath slow a little more. Let the thinking, if it continues, become background rather than foreground. You are in the recovery phase now. You have been here before, in sleep, every night, and you will find it again tonight. The recovery from chronic stress is not dramatic. Research consistently shows it occurs gradually, in small increments. Improved sleep one night, slightly lower cortisol the next morning, marginally better emotional regulation over a week. The body does not announce its healing. It does it quietly. Underneath the ongoing demands of life, in the hours of sleep and stillness that are allowed to accumulate, recovery is less a destination than a direction, a slow drift back toward equilibrium that the body is always attempting when given the chance. The glymphatic system, a brain-wide waste clearance network primarily active during deep sleep, removes metabolic byproducts, including beta amyloid and tau proteins that accumulate during waking hours. Stress disrupts deep sleep and therefore impairs glymphatic function. This connection has drawn interest from researchers studying neurodegenerative conditions. The evidence for glymphatic activity in humans is still accumulating and requires careful interpretation. Much of the foundational research was conducted in rodents, but the sleep-waste clearance relationship represents one of the most compelling, emerging explanations for why sleep is so non-negotiable for long-term neural health. NREM sleep is where the brain rewrites its physiological stress signature. During slow-wave sleep, cortisol reaches its daily low point. Growth hormone is released. Tissue repair is initiated. The immune system consolidates its defenses. Every one of these processes is an active form of recovery from the stress the day produced. The night is not passive. It is the day's counterpart, the biological answer to everything the waking hours asked of the body. Chronic stress reduces the complexity of social behavior. Research has found that individuals under sustained stress show reduced empathy, lower patience in social interactions, and narrowed social circles over time. This reduction is not a character change. It is a resource allocation change. The stressed nervous system conserves its limited regulatory capacity by withdrawing from the cognitive demands of complex social engagement. This is why reconnecting with others and feeling genuinely understood is not merely pleasant during stressful periods. It is restorative at the level of the nervous system. The relationship between music and sleep has neurological underpinnings. Music with tempos near 60 beats per minute aligns with resting heart rate and promotes entrainment. The tendency of biological rhythms to synchronize with external rhythmic patterns. Slow music during the pre-sleep period has been associated with faster sleep onset, reduced nighttime waking, and subjective improvements in sleep quality across multiple studies. The ears never fully close during sleep. The sleeping brain continues to process auditory input, particularly for sounds with emotional significance. What fills the room at sleep onset genuinely shapes what happens within it. Stress and creativity are linked through the same neural systems that mediate the stress response. High amygdala reactivity and heightened default mode network activity, both associated with chronic stress, can also, under certain conditions, contribute to associative and imaginative thinking. Some of the most creatively productive periods in human history have occurred under conditions of substantial external stress. This does not justify stress or romanticize suffering, but it acknowledges that the human capacity to make meaning, to create and imagine, is deeply woven through the same systems that process adversity. And that rest, in restoring those systems, restores the capacity for both capacity. Research on the neurobiological effects of tea, specifically L-theanine, an amino acid found primarily in green tea, has found that L-theanine promotes alpha brainwave activity, reduces cortisol levels in stressed individuals, and diminishes the physiological stress response to demanding tasks without causing sedation. It appears to work partly through interactions with GABA receptors. The same receptor system targeted by many pharmaceutical anti-anxiety agents. A warm cup of tea in the evening, then, is not merely cultural ritual. It is a mild but real modulation of stress neurochemistry. One humans have relied on, without knowing the mechanism, for thousands of years. Sleep spindles. Brief bursts of oscillatory neural activity during NREM stage 2 sleep are associated with memory consolidation and the protection of sleep continuity against external disturbances. Research has found that stress reduces sleep spindle density. This reduction contributes to more fragmented sleep and impaired memory processing. Interestingly, sleep spindle activity is also associated with GABA, ergic inhibition in the thalamus. The same inhibitory signaling that calms arousal. The quiet inner oscillations of the sleeping brain are, themselves, a form of neurological rest, the brain's own method of settling itself through the night. The act of naming an emotion, what neuroscientists call affect labeling, reduces amygdala activity in response to emotional stimuli. Research by Matthew Lieberman at UCLA using functional imaging found that simply putting feelings into words produced measurable dampening of the brain's threat response. This is why talk therapy, journaling, and even conversations that give voice to what is being experienced have neurobiological effects beyond what they feel like subjectively. The mind that can say, "I am anxious," is neurologically in a slightly different state than the mind that simply experiences anxiety without naming it. The brain's response to uncertainty is, in many respects, more stressful than its response to confirmed bad news. Researchers found that the anticipation of a possible negative outcome produces more sustained cortisol elevation than receiving a confirmed negative outcome of equivalent magnitude. The reason appears to involve the brain's need to maintain all possible response preparations simultaneously under uncertainty. Resolved situations, even negative ones, allow the brain to select a single response strategy and begin recovery. Unresolved uncertainty keeps the stress system in a state of readiness that cannot yet shift into repair. Research on post-sleep cognitive reappraisal, the ability to think about stressful situations differently after a night of sleep, has found that individuals who slept following exposure to stressful material were significantly better able to generate alternative interpretations of the same situations the following morning. The improvement was correlated with the proportion of REM sleep obtained. The night's processing does not merely reduce emotional intensity, it appears to increase cognitive flexibility around the very situations that felt most fixed. Morning perspective is not simply distance. It is, in a neurological sense, a genuinely different view. The heart and brain communicate through both neural and hormonal pathways, cardiac coherence, ordered, rhythmic heart rate variability associated with calm, has documented effects on prefrontal cortex function. Research from the HeartMath Institute, alongside independent biofeedback studies, has found that rhythmic breathing practices reduce cortisol and support emotional regulation. While some HeartMath findings await broader independent replication, the underlying principle that rhythmic breathing modulates autonomic tone is well supported across the wider literature. The heart is not simply responding to stress, it is participating in the regulation of it. Research on longevity has found that perceived chronic stress is among the most consistent psychosocial predictors of earlier mortality, operating through its effects on cardiovascular health, immune function, inflammatory markers, and cellular aging. This finding appears across diverse populations and study designs. It is not presented here to add another source of worry, which would be precisely the wrong conclusion to draw. It is presented because it underscores the biological seriousness of rest, sleeping, recovering, protecting one's stress physiology. These are not self-indulgences. They are, in the most literal biological sense, life-sustaining acts. The specific pattern of breathing during slow-wave sleep, slow, regular, deeply diaphragmatic, is one of the strongest activators of the parasympathetic nervous system available to the body. Deliberately breathing this way during wakefulness, slowly from the diaphragm, with longer exhalations than inhalations, mimics this sleep-associated pattern and produces measurable parasympathetic activation. Each exhalation is, neurologically, a moment of surrender. The body has always known how to rest. The breath, in this sense, is the path the body takes to get there, available in any moment, requiring nothing more than the willingness to slow down. Daylight, natural, outdoor light, especially in the morning, is one of the most powerful regulators of the circadian system and the cortisol rhythm. Morning light exposure anchors the cortisol awakening response, stabilizes the timing of melatonin release in the evening, and strengthens the circadian signal that governs when the body is ready to sleep. Research has found that individuals with greater morning light exposure show better sleep quality, lower evening cortisol, and more stable mood across the day. The simple, ancient act of moving into the light in the morning is, physiologically, one of the most effective preparations for the rest that will come that night. Research on digital technology use and stress has found that the constant availability of information, communication, and demand through smartphones is associated with higher baseline cortisol levels and reduced heart rate variability. The anticipation alone of possible incoming messages, even when the phone is face down and silent, produces measurable cognitive and physiological arousal. The device associated with so much of modern life carries, in its presence on the nightstand, a low-level signal of potential demand. The simple physical distance of the phone from the sleep environment has been associated in research with improved sleep quality and lower pre-sleep anxiety. Cognitive behavioral therapy for insomnia, CBT, is the first-line recommended treatment for chronic insomnia by multiple major medical bodies, including the American College of Physicians. Its effectiveness is grounded in addressing both the behavioral patterns and the cognitive beliefs that perpetuate insomnia, including the worry about sleeplessness that itself maintains arousal. Research has found CBTI to be more effective than sleep medication for long-term outcomes. Without the risk of dependency, its core insight is that the beliefs and behaviors built around sleeplessness often sustain it more than the original cause. Understanding sleep, in this framework, is genuinely therapeutic. The sleep experience of feeling rested is not simply about duration. Research on sleep quality consistently finds that subjective sleep quality, how rested the body feels upon waking, is more predictive of daytime stress resilience, mood, and cognitive function than total hours logged. A night of uninterrupted, slow wave-rich sleep of 6.5 hours can leave the nervous system more prepared than 8 hours of fragmented, stress-disrupted sleep. This means that the environment and the physiological conditions of sleep matter as much as its length, perhaps more. For many of the people carrying the most stress, research on the stress response in animals and humans has found that the mere presence of a familiar, trusted individual is sufficient to attenuate the cortisol response to a threatening situation, a phenomenon called social buffering. This effect does not require verbal communication or active comfort. Proximity to a trusted presence is physiologically calming in itself. The body carries this biological need for co-regulation. The nervous system is, in evolutionary terms, designed to be regulated partly through connection with others. The isolation that so often accompanies stressed periods is not just emotionally difficult. It removes one of the body's primary biological mechanisms for recovering from the stress itself. The experience of awe, a sense of vastness, and the need to accommodate something larger than one's current conceptual framework has been found to reduce self-referential worry, lower inflammatory markers, and expand the sense of available time. Research by Dasha Keltner at UC Berkeley found that awe-inducing experiences reduced the dominance of the default mode network's self-focused processing. The mind, for a moment, becomes more interested in what is outside it than what is inside. And in that shift of attention toward the vast, the beautiful, the incomprehensible, the stress that lives primarily in the self-referential mind grows. For a time, quieter. Among the most consistent findings across the entire field of stress research is this: sleep is not a passive state from which the stressed body is merely absent. It is the primary biological mechanism through which the stress response is metabolized. The brain is restored, the immune system is maintained, and emotional experience is processed. Every hour of sleep is doing something. Every night of rest is an act of repair. The body has always known this. What science has done is make the language of that knowledge available to the mind as well. Stress resilience research has identified the concept of benefit finding, the capacity, after difficulty, to identify what was learned, strengthened, or clarified, as associated with lower long-term stress reactivity and better health outcomes. This is not the same as toxic positivity or denial of difficulty. It is the brain's engagement of the prefrontal cortex in a search for meaning within adversity, a cognitive process that appears to genuinely reduce the amygdala's sustained reactivity to the same memories. The mind that can hold difficulty alongside meaning is not a naive one. It is, in a neurological sense, a more regulated one. The same neural circuits that process stress also process meaning. The prefrontal cortex, the region most suppressed by chronic stress, is also the region most involved in the construction of narrative, purpose, and long-term perspective. In restoring the prefrontal cortex through sleep, through rest, through managed stress and social connection, we are not merely reducing cortisol, we are restoring the neural capacity for the very things that make the stress feel worth bearing. The clarity to understand what matters, the perspective to hold difficulty without being consumed by it, Research on the neuroscience of hope has found that hopeful anticipation produces distinct neural activity in the ventromedial prefrontal cortex and anterior cingulate, and is associated with reduced amygdala reactivity to negative stimuli. Hope, in this neurological sense, is not denial of difficulty. It is an active regulatory state, one in which the brain is oriented toward the future in a way that dampens threat-based processing in the present. The nervous system that can hold the possibility of rest, that can allow itself to move toward sleep rather than resist it, is practicing, in small form, what the science of hope describes. Tonight, science has named what the body has been carrying. The stress that activated the amygdala, elevated cortisol, and kept the mind turning. It has a name now. It has a biology. It has pathways that lead not only into activation, but back out of it. Can understanding stress science actually help your brain let go of it? The science says yes. And perhaps something in this moment says yes too. When science names what your body has quietly carried, the nervous system finally has permission to rest. You've traveled far tonight, through the amygdala's ancient alarm, through cortisol's quiet persistence, through the neurons that fragment sleep and the slow-wave cycles that restore it. You learned that a single sleepless night raises anxiety by up to 30%, and perhaps that knowledge itself loosens something. You heard that deep sleep is the brain's natural anxiolytic, its built-in mechanism for softening what the day made hard. You didn't just hear science tonight. You gave your nervous system a long, unhurried explanation of itself. And somewhere in that explanation, something may have begun to settle. Stress is not the enemy of the life you are trying to live. It is a signal from a body that cares about that life deeply. When science helps you understand it, the signal loses some of its urgency. And in that softened urgency, sleep, which was always waiting, finds its way in. One sleepy soul at a time. Let the facts dissolve now. Let the understanding soften into feeling. Notice the body, heavy, held, breathing on its own. The nervous system that has been listening all night is already calmer than when you began. Not because anything has changed in the world, but because something has shifted in the space between you and what you carry. You can let it rest now. All of it. Breathe out, and let the night do what the night was made to do. If this thoughtful exploration of the mind helped you relax or find some stillness tonight, a gentle like and subscribe helps other sleepless souls discover this peaceful space too. It only takes a moment, but it means the world to us. One quiet tap, one sleepy soul at a time. But there's no need to do anything now. Nothing at all. You've traveled far tonight, farther than most journeys take you. You can allow these thoughts of stress and the quiet science of recovery to fade now, like the last dim signal of a system finally standing down, leaving only the soft, weightless calm behind. Let your breathing deepen. Let your body feel heavier, as if the earth itself were gently pulling you into its patient stillness. If sleep comes, let it carry you like a current you have finally stopped resisting. Smoothly, inevitably, into a place where time and weight no longer ask anything of you. If your eyes are still open, the next video is waiting, ready to continue the journey. But if they're closed, stay there. Stay in the darkness that feels safe and warm. Tomorrow, the cortisol will rise again, and the world will make its ordinary demands. But tonight belongs to stillness. Rest well, quiet mind, and remember, the nervous system that learned to hold all that tension also knows, somewhere beneath everything it was taught, how to let go. Where science meets sleep, dream deeper with bedtime science. Good night.